Hi, everybody. Good morning, good afternoon. It depends on where you are. My name is Aline, and I am a data scientist working for Knime. And today I'm having the big pleasure of joining Professor Dayajan Vijayasinha, known by us as Professor Shanaka. He's an associate professor of pharmacy at Virginia Commonwealth University, and he has been collaborating with Knime for a long time now, and especially in the creation of use cases for digital healthcare. And today, two of his students that he will introduce to you will present some of these use cases that we actually compiled in a booklet that we will also talk a little bit about later in this webinar. So thank you very much for being here, and I will pass to Shanaka.
Thank you, Alini. I am going to, I was initially going to do this, but I think I'm going to share the screen. Can you guys see it? Great. Welcome, everyone, and thank you for joining us today. This started off more as an experiment, and now it has snowballed into something that has practical value, and not just in the US, but more along the lines of globally. And I'll give you a few thoughts on how we came up with this idea and why we moved forward with it. And as always, the proof is in actually what students were able to do with it. And these two people here will explain that much better than I can, right? So first thoughts, right? Early observations in educating health professionals in digital health. These are the observations that led to this whole program. What we saw was that healthcare professionals are the best suited for identifying use cases for digital health interventions. In the sense, when they're practicing, they see, well, we have this data. It would be great if we can predict this outcome from that data, right? It's not something that we can easily tell someone else to do it, but because these students are actually immersed in this environment, they understand it really well. So when they see the data, they also intuitively know the sequence of steps that they need to take in order to transform that data to gain what is called practical insights. You can gain insights from any data, but are they practically useful insights? It's only the healthcare professionals who will know that the health data that they transform, what they get at the end is a practical insight. These kids also have the clinical background to test the accuracy of the insights. And finally, they have the network of colleagues. You can build something and you're finding it, oh, this is really useful in my clinic. I wonder whether it's a case in other clinics. So they have this network of colleagues that they can share their workflows with, share their application with, to try and test it in multiple different settings. Then, okay, so after that observation, you're like, okay, healthcare students need to learn how to build digital health application. Now, how do we do this? Do we take Python? Do we take C? Or do we take a Java approach? Well, none of this will work because these guys, their curriculum is completely packed. They don't have time to learn programming, right? So the kind of things we need is a no coding platform, right? It should be open source because university might have a license to license for a complicated program, but if they graduate, once they graduate, they don't have access to it, then they cannot continue learning. It would be great if they have standardized learning modules, and Knime actually has L1, L2, L4, and so on, right? Workflow should be easy to comprehend because they should be able to show it to another colleague and say, look, these are the steps that we followed, and this is what we did, right? They should be able to understand it without having to understand actual coding that goes into it. They should be able to create interactivity. They should be able to create reports. It should be easy to convert into web applications if they want to deploy it across a hospital setting, and allows functional prototyping, right? So the platform that had all of that was Knime for us, right? This is like a whole lot of things that have to come together in a single platform. And I've been doing extensive searches, and Knime was the only thing that had all of these for us. And how did we train the students? We started with an extracurricular approach. In the sense, we created pharmacies for digital health that allowed students to learn outside of their classroom how to develop digital health applications using Knime. Then we went to a 2-credit elective that Sebastian took, right, among other students, Introduction to Data Science and Rapid Prototyping, where they learn over a semester how to create digital health solutions. We had a research elective, and I don't remember whether you took it, Sebastian, but some of the students move on to take the research elective, and then the advanced pharmacy practice elective, which is again something Sebastian did, right? And then we created a playground that allows them to learn collaboratively and to practice skills. So Knime already did a lot of work for us. They had the self-paced learning, and it's an absolute requirement that they complete L1 and L2. And with just those 2 levels, students can create digital health applications. We needed health-related data science projects. Yeah, sure. Personalized pharmacotherapy calculators that we presented a while ago was one of those, but there's plenty of applications that we can create solutions for. And these are some of the use cases, the first 3 use cases of the API rotation, advanced pharmacy practice elective rotation. And I'm not going to go into detail. These are already out there in the Knime digital health space. But I want to give a little bit of an introduction to the 2 students here. Sebastian, who's going to be presenting first, is a PharmD candidate. He'll be graduating in May. And him and his team has created an amazing digital health application, and he continued using Knime, and then he continued on to create additional applications in digital health. Lama, on the other hand, already has a PharmD, and she's doing her PhD now. And she's created this amazing application for post-market surveillance using Federal Adverse Event Reporting System. So I don't want to take too much time. The students actually did all the work. So I'm going to hand this over to Sebastian. All yours, Sebastian.
Awesome. Thank you so much. And so today's conversation, it's really going to be a conversation and me telling a story about how I became one of Shanaka's kind of testing students or testing experiments and went through. And so I was put in with a team of about 7 people, and we took the initial class of digital health. And I was presented with this challenge by Shanaka being like, take any issue or anything that we can optimize within pharmacy and create it with Knime. And so after going through the learning modules, the L1 and L2 learning, then that kind of gave us a little bit of the tools to be able to kind of go through this. But I kind of wanted to kind of touch upon our entire learning process. I want to briefly show you the final product first, but then kind of show you the path of how we got there. So I'm going to quickly share my screen and show you this calculator. And to give you a very, very brief background, there's a topic about coagulation where your blood clots, and that can lead to different disease states such as deep vein thrombosis or DVTs or PEs, and which is a pulmonary edema. And so these are blood clots that are developed throughout the body. And we use this medication called heparin in order to kind of dissolve that blood clot. However, using this medication, it's an IV bag, and you basically kind of titrate, meaning you give a specific amount of the medication at specific rates, specific doses, and you adjust the amount of medication that you give depending on something called the APTT, or activated plasmin prothrombin time. And so basically, this is a measure that it's basically how fast your blood will coagulate, to keep it simple. And so this is a very, very simple application that it looks simple in overall, but there was a lot of work put into it. And so I kind of want to quickly dive into the story of how we got there. When Shanaka presented us the idea of take something that was difficult and make it simple and automated, we immediately thought towards our class of pharmacokinetics, and we basically learn all these equations and apply them to measure how a drug goes through the body and interacts with our body through a kinetic standpoint. And we're worried about memorizing so many drugs, so many interactions, and so much. And not all of us like doing this manual task, and a lot of the calculations takes a lot of time. And so we initially started kind of exploring this idea of implementing or using Knime to kind of go through different equations. And so I have a couple of pictures here where we started kind of all brainstorming. We started writing down all of the equations that, thinking back of different test questions, what could we just easily automate so we don't have to do these equations ever again? In the end, it was just like a huge conundrum of like, what equations should we use? How do we implement it? And keep in mind, we have no coding experience. We have no idea how to really approach this, since we're not really taught in school how to think about a logical kind of stepwise process for data transferring or data kind of thought process. And so, we were like, all right, how can we really create a project out with using Knime? And we were taking our cardiology class, and we came up with the idea of, what about using heparin nomograms? And to quickly explain what a heparin nomogram is, it's basically kind of like a step-by-step guide on how to dose heparin. And all hospitals have a heparin nomogram. They don't really have a calculator that they've implemented within their EHR systems or whatever systems they use. And so they manually have to take a look at whatever rate the heparin is running at, take a look at lab values, aka the APTT. And then based off this APTT, then they have to manually do the calculations and adjust and go through all of those steps. And so we were like, what if we just took this and just simplify it, make everyone's lives easier, right? So what we came up with was our heparin calculator. But before I go into our Knime process, I kind of want to show you how we actually designed this on the whiteboard. We kind of designed this as, we have a specific input, then we have these set of rules where all the calculations happen, and then you have whatever text comes out. It's very, very simple. You just input something and then you get something out. You miss the complete kind of calculation step. And that's what we really try to aim with doing with Knime. So now I want to touch upon specifically the program that we built. There's 3 different calculators. One's for general cardiovascular, and another one is specific for DVT and PE, and another one is specifically for the pediatric population. However, for this conversation, I'm only going to be talking about the DVT PE, since everything else is very, very similar, just certain calculations are different. And so within this component, we were able to kind of divide things up into kind of the initial bolus or the initial infusion rate, meaning what is the first kind of amount of drug that you give and at what rate. And then we divided up the second portion of the calculations into kind of the adjustments that you do after you've given that initial kind of rate. And so going into the initial bolus and also initial rate, it's a very, very simple kind of logical step-by-step processing that goes on. We start off over here with all of the inputs where we kind of put in the patient's weight, put in the formulation of the heparin product that you're using, and also you put in the weight of the patient. And so this just undergoes data processing, where you're just doing basic math formulas to convert, weight and also formulation and how many mLs you're actually getting into something that you're actually able to see. And over here, this tile view allows you to basically see whatever calculation comes out of it. And so over here, you're able to see what it actually looks like from the actual program standpoint. You get to input the patient's current weight. Over here, this was kind of like a challenge for us, being able to input like a button to convert from kilograms to pounds and just seamlessly implement it within the program. In hospitals, you only use kilograms. But anyways, we also have like all of the heparin formulations over here, and you get to input all of these. And all it does is it's a quick, simple calculator that just tells you exactly what to do. And this is fantastic and all, but then it came the challenge of how do we go about and adjusting. And I quickly want to transition back to this PDF where, after you do all of the initial infusions based on a weight-based dosing, then after you've gotten your APTT baseline, then you have to start adjusting. And so there should be adjustments done every 6 hours or every 4 hours, depending on how your APTT is. And if your APTT is low, then you're at risk of coagulation, meaning that a blood clot there's a higher risk of developing a blood clot. And then if it's very high, then you're at risk of bleeding, and that leads to brain hemorrhages and many, many, many other issues. And so keeping a very tight control is very essential, and this is why critical drug that it needs tight monitoring. And so over here in this nomogram, you're going to notice how you have to be able to input a specific APTT, and then that value will kind of shift you towards going through one direction, right? So if your APTT is anywhere between 40 to 54, then you have to adjust the bolus by 1,000 units. You don't have to stop the infusion time, but then you have to increase you have to do a dose adjustment and then in terms of the rate, and then repeat at a specific time. However, once going towards like a very far end, if you have an APTT over 150, you don't have any calculations or any numbers. You actually have a set of directions that like you want to tell the provider. And so a big challenge was, how do we get the program to kind of tell us a set of numbers? And then specifically, once you input a specific APTT, how do you actually get it to give you a set of instructions? And so that's kind of what we did over here. I'm going to actually shift over to the post APTT adjustment program, and it's big. This is kind of like what we came up with, and I'm just going to quickly zoom out. All of these kind of lines represents a different APTT kind of, logical adjustment based off the nomogram. You have your inputs over here, very logically set. Inputs, processing, outputs. And then over towards the bottom end, which I'm going to explain a little later, it's kind of where we actually developed the entire kind of tech, what actually prompts the program to give you text. And I quickly want to do this comparison of this page with our actual kind of diagram. It kind of came a little close, and so it's very easy from a non-coding standpoint to just put something on a whiteboard, try to logically and systematically kind of link each node together, and then have something that will output whatever you're trying to do. So quickly going through it, we have all of our inputs over here where we input our APTT, current time and military time, selection of concentration, and then the infusion rate. We have all of these, these different nodes. And when I was learning how to use Knime, I really kept it simple. I always thought connect all of the circles to the circles. Then this variable to table node, table row node collects the circles to the triangles. And that was honestly a great starting point for me to actually start developing this with my team. This goes through all of these switches and all of these kind of processing nodes where you can do math. It basically, you're able to implement rules of, if the APTT is within this range, then display this text or display this number. And it really goes way beyond that. And towards the end, we were able to initially input or like have a program that just gives you numbers. But then came the challenge of, how do we change from like numbers to actual text? AKA, what happens when your APTT is greater than 150? One of my very good friends, Amir, he actually came up with this genius idea. And keep in mind, we were not Knime experts, so we were figuring everything out like on the spot. He figured out that, let me quickly change this to 160. Oh, it's currently at 160. Give me 1 second. Let me quickly change this to, let's say, 167, and then run this. So he figured out that you can actually create this you can trick for us, we call it like tricking the program. Once it kind of goes towards the 150 range, we couldn't really find out how to input text. And so we created this constant value column where we kind of, any value or anything that goes through this path gets converted to a string value of something called NA. And after you determine that string, you're able to filter everything out through a row filter of basically including everything that is called NA. And we're able to use something called a table creator to just input all of our text, and that automatically gets rid of all of the numbers that would otherwise come through with the workflow, and everything gets displayed by this table view, which is just all of the text that you actually get to see. And so I quickly want to finalize our conversation today by actually showing you kind of the second portion over here, where I'm actually going to change this back to 100. And just for, let's do 1400 and an infusion rate of 4, right? It basically goes through all of the steps, and it tells you exactly what you need to do. All of this information is very easily available. However, the step that we were trying to optimize is the entire workflow and kind of thought process behind it. It tells you exactly what time you have to repeat your measurement in military time, tells you whether you need to do an adjustment bolus dose, tells you if you have to adjust any rates, and then it also tells you, if you have to stop it or not. And I quickly want to show you real quick what happens if you go a 150 and above. It will tell you exactly kind of like what the steps you should be following. And it kind of tells you right there, attention, please follow these prompts. And you can use basic HTML to do all of the texts over here too. And that was basically it. But it was really, really kind of like a big learning experience, because we don't have any coding experience. We have a lot of clinical knowledge, but how do we create something that's useful for providers? How do we create something that's useful for people? And it's easy, how Shanika always said, it's easier to teach a clinician how to code than a coder how to do clinical stuff. And using a non-coding format, it really kind of worked out for us. And a lot of providers do find this tool useful, since it really maximizes time. It saves, people in critical settings, such as an APTT greater than 150. And it was overall a great learning experience. And that's basically it. Towards the end of the presentation, feel free to ask me any questions. But it was a great journey, honestly. And I'm going to pass it off to Lama now, so she can share her journey with this.
Thank you, Sebastian. That was really awesome. Okay, so I'm sharing my screen now. Hi, everyone. My name is Lama. I'm a PhD student at Virginia Commonwealth University. So, and I'm happy to share with you my master's graduation project, which I worked under the supervision of Dr. Shanika, which is about the identification of adverse drug events from FAERS database by using the low-code analytical platform called Knime. So, okay, so during my master's journey, I was really interested to undertake research aimed at providing a unique perspective on drug-related adverse events by incorporating skills on data science such as Knime. Also, there is a need for the post-marketing surveillance, or let's call it the PMS. So post-marketing surveillance refers to the practice of monitoring safety and effectiveness of pharmaceutical drugs or medical devices after being approved to be used by regulatory agencies. So the PMS, or post-marketing surveillance, are used for detecting rare adverse events, assessing the long-term effect of medication, represent a wider population, and capture the effect of the real world. And lastly, it identifies the interactions, whether they were synergistic, which means a positive interaction, or if they were negative or antagonistic reactions. And that's the main aim of this project. And accordingly, my interests align actually with the objectives of FAERS. So what you see here, this is a screenshot of the FAERS. It represents the FDA Adverse Event Reporting System. It is a public database maintained by the US Food and Drug Administration, and it contains an information on adverse events and medication errors reported to the agency. And this database was actually established to provide a centralized system for collecting and analyzing reports of adverse events related to the FDA-regulated products, including drugs, biologics, medical devices, and dietary supplements. So the FDA adverse events, or let's say the FAERS, it actually identifies the potential safety concerns to monitor the safety and of the safety of the drugs and biologics, and to inform regulatory decisions. And also these regulatory decisions are used to support the post-marketing surveillance activities, such as epidemiological studies and risk management activities. So to sum up, the FAERS is used to identify potential safety concerns, assess the effectiveness of post-marketing surveillance programs, evaluate drug utilization patterns, and generate hypotheses for further clinical investigations. Now, although the FAERS is a very powerful tool to capture the safety signals, but there are several limitations to this database that should be taken into account. So first, it has a self-reported nature. That means the results might be subjected into bias, and the timing of the adverse event or the report is not mentioned. Plus, it has an underreporting nature by physicians. Additionally, reports may be inaccurate, resulting in an unsupervised collection of results that may contain duplicate or incomplete information. And one of the major challenges is that in terms of deduplication and mapping. And that's why clinicians, researchers with low coding expertise, they send their data into research centers. And these centers must have a comprehensive understanding of the data structure, and they have to know how to interpret these data in order to have an accurate result and to have a greater impact. Another actually difficulty is that the structure of this data is very difficult, that it needs a person with a very sophisticated computational skills to extract relevant information. And usually clinicians do not have that skill. And it is a very time-consuming process. So according to these limitations, we actually developed a pipeline to extract, to standardize the FAERS data by using Knime. So Knime, it's an open source analytical platform that allows the users to build and deploy data science workflows, including data pre-processing, analysis, modeling, and visualization. It has a lot of advantages. It's an open source, everyone can use it. It is very easy to use with just one click on the node itself, or to execute the node, the workflow will automatically generate the information. It allows for collaboration, and it has a community support called the Knime Hub, where you can post your question or your workflow and you will receive answers within minutes. So this platform is used to integrate data from various resources such as databases, spreadsheets, and cloud services. And it has a pre-built nodes. Again, you don't have to have sophisticated computational skills or coding expertise to work with it. And lastly, when we actually used the Knime to download the files from the FAERS database, it took us around 7 hours, while compared to when we extracted them from the data itself, or sorry, from the database itself, it took us around 1 week. So it is a very time-efficient process or platform. Now, before I tell you how did we extract and standardize the data, let me tell you that not only this workflow is not only used for this purpose, but we can actually use this workflow to address the question whether the concurrent use of a drug can reduce another medication-related adverse event. So let's suppose that drug X has a specific adverse event, and we want to see if drug Y can decrease the drug X related adverse event. So by using this workflow, we can actually generate the groups and to compare these adverse events with and without drug Y. So let me show you the process. We first downloaded the files from FAERS, and after that, we've loaded them into Knime and removed any missing values, and we removed any duplicated reports. Then we created a nomenclature harmonization for the drugs, for the adverse events, and for the diseases. Then we've standardized the rest of the FAERS files. And after that, we were able to generate the groups and compare the adverse events with and without drug Y. So let's see. The first step is we've downloaded these files manually from the years 2015 to 2020. And the reason behind choosing that gap or that timeline is because we wanted to test that workflow at first. And then these files were loaded into Knime, and yes, so these files were loaded into Knime, and then we've combined all of these files into one component. We call it the meta node, which is, I renamed it as the FAERS processing. Now, inside the FAERS processing meta node, you can see this whole workflow, and we've actually worked on 4 types of files or tables, and they are the demographics, the drugs, the indications, and the reactions, which I will address each of them separately. So the first thing, we've looked at the demographic table, and we've noticed a few things. As you can see here, the age and the weight, they are expressed in different units. So we wanted to create standardized units. And the sex or the gender column is expressed in male, female, so we wanted to convert them to binary values or vectors in order to make it easier for clinician researchers or the user to develop their own analyses. And then we wanted to convert the occurrence country into regions. Now, the whole purpose of doing that is we want to create an amenable data set for a person with low coding expertise to look at the data to extract relevant information for their own use. So the first thing we did is, after following the file path, we've used that table row to variables to convert a single row to a different set of variables to make it easier for the user to read it using CSV Reader. And then we began by selecting columns for preparation, and then the data was further filtered by removing missing values, by applying a set of rules, keeping those that matched the rules, and removing those that did not. And then we moved on to the regions. So for the region, we did the following. We've used a reference table that has the regions that we are interested in, and then we've used the reference row splitter. So this node actually is used to modify the original table according to the information provided from the reference table, and then we've assigned constant values for this region. As you can see here, on the left is the table or the column before the adjustment, and on the right, after the adjustment. And then our next step is that we've used the rule engine to convert the male and female into numbers. So 0 for male, 1 for female, and 2 for unknown. And then lastly, we wanted to convert the weight from pounds, or we wanted to create standardized weights into kilogram. And we use a specific age schema to convert all of the ages into years. And as you can see, the table on the right is the adjusted one. Now, this is the final table of the demographic after doing our adjustment. And then let's move on to the drugs. Let's say that the user is interested in drug X and drug Y. So the first thing we did is we want to create groups according to the medications the patients were taking. So group 1 will be for patient taking drug X alone. Group 2 will be for patient taking drug X and drug Y. And group 3 for patient taking drug Y alone. But before generating these groups or creating these groups, we wanted to extract the drugs of interest from the FAERS. So usually the medications or the drugs, they appear in upper or lower cases and may include generic and brand drug names and also symbols. So what we did is that we wanted to make sure that we've extracted all of the drug names using the regex find all. So let me show you what we did. So inside the generate group meta node, we've used a reference table that has a generic and brand drug names of interest, and then we've extracted them using the regex find all. So this node creates a digital list that capture all the drugs name. And then we counted the number of strings that match the queried drug and removed the non-captured drug. Now, this is the first step after extracting the generic and brand drug names of our medications of interest. The next thing is we want to generate the groups using the column expression. So usually the user or researchers, they look at group 2, which indicates that the patient is receiving both of the medications X and Y. Just one information, let's say that the user or anyone, they want to change the drug names. They can actually manually double click on that node and they can manually change the drugs, and the workflow will automatically generate the groups according to the drugs of interest. So you don't need to own sophisticated or coding experiences. It's very easy to use. Now, our next step is we want to see if there are other medication than drug Y that decreases the drug X related adverse events, because we were really interested to see if these adverse events are decreased by drug Y or by other medications. So what we did is the following. We've used a reference table that has the generic and brand drug names of these medications that may decrease the adverse event. And then we've extracted them using the regex find all, and we've assigned constant values for them to be 1 if the patient actually took this medication and 0 if the patient did not take them. So this is the final drug table. As you can see here, we did a lot of adjustment. So we've created the group column, and then we have the generic and brand drug names of the drugs, and then we have the constant values. So we will always look at group 2. That means the patient is receiving both of these medication X and Y, and you have all of these information provided. Now, let's say that you are interested to explore a specific disease. Well, we can do that. So inside the indication meta node, we provided a reference table that has the reference table that has the disease of interest, and we've extracted them using the regex find all, and then we've assigned constant values to be 1 if the patient developed or has the disease of interest, and 2 for all of the other diseases within the FAERS file. So again, if you are interested in other diseases other than cancer, you can actually manually enter the names, for example, hypertension or dyslipidemia, anything, and the workflow will automatically generate the groups according to the drugs of interest and according to the disease of interest. Now, this is the final disease table. As you can see here, we've did a lot of changes. So the first thing is we created a new column called the primary indication, and then we've collected all of the indications in one row instead of putting them in different rows. Now, lastly, we're now interested in the adverse event or a specific adverse event. So we did the following. We've used a reference table that has an adverse event of interest, and then we use the reference row splitter to apply or to extract the information from the reference table. And then we have the constant value columns, or we gave a binary values to be 1 if the patient developed that adverse event and 0 if the patient did not develop it. Now, this is the final table on the right after our adjustment. After we finished these 4 types of files, we wanted to do something. So we wanted to create data harmonization for nomenclature, and this will help in sharing the information internationally. It is basically having a language that everyone understands. So we did the following. We followed a similar approach used in this article. So this article is used or did a data harmonization, but they used a very sophisticated coding language, which was not the aim in this project. So what we did is that we applied their standardized vocabularies for the drug names, indications, and the reactions by downloading them from the Athena database, which is used for data harmonization. And after that, we've uploaded them into Knime, and we've joined the concept unique IDs with using the joiner node with the drugs, indications, and reactions. Now, this is the final table. Again, the purpose of doing that is we want to create a standard medical terminology in order to share this information internationally. The last step we did is we want to do a bit of cleaning. So we cleaned the data by removing any missing values, renaming few columns, and provided a node that allows the user to download the result on an Excel file. Now, let me show you the final table. So this is actually, the table is bigger, but for the sake of this presentation, I want to show you the main points. So now let's say that we are interested to see if drug Y can decrease the drug X related adverse event. The first thing we will look at is group 2, which means that the patient is receiving both of drug X and drug Y. And then we have the demographic information of that patient. And then we have the column indicating that the patient did not develop the reaction of interest, and maybe because the patient is taking other medication than drug Y that decreases this adverse events, and then the patient has the disease of interest. Now, just to sum up before we move on to the next slide, the purpose of doing that is we want to create an amenable data set for the user with low coding expertise to look at this data, to extract relevant information, and to build their hypotheses or to do post-marketing surveillance. Now, the next step is we wanted to do or to try this workflow on a real-life medications. So we collaborated with clinicians, and they were really interested in the story of bevacizumab and aspirin. So bevacizumab is a drug used for cancer treatment, and it has been associated with high blood pressure or hypertension as an adverse event. So studies suggest that a low dose of aspirin might decrease the bevacizumab-induced hypertension. So we wanted to test that. And this is the workflow that we use to test that hypothesis or to test that information. So again, we just did the selecting files for preparation, removed any missing values, we've assigned the regions, and then we've converted the male and female into vectors, and then we've converted the weight into kilogram and age into years, and this is the similar demographic table. And then the next step is we want to generate the groups. So group 1 will be for patient receiving bevacizumab alone, group 2 for patient receiving bevacizumab and aspirin, and group 3 for patients receiving aspirin alone. So we wanted to extract these medications first, the generic and brand drug names, and we wanted to create the groups. And in the second and third component, so we wanted to see if other antihypertensive medications can decrease this hypertension and not the aspirin. So we've extracted them and we've assigned constant values for them if the patient took them or received them or not. So this is the final table for the drugs. And lastly, because bevacizumab is used to treat cancer, so inside the indication component, we've extracted the patient having cancer and assigning a vectors to indicate the diseases. So 1 will be for patients who have cancer and 2 for all other indications within the FAERS. As you can see here, this is the table for the indication. And then we are interested in hypertension or high blood pressure. So we've used a reference table that has the high blood pressure and extracted them and assigned a constant values for them to be 1 if the patient developed hypertension and 0 if the patient did not develop it. Now, after doing all of this, we've joined all the tables into one table. And now we want to see the results. So for data visualization, we've extracted the hypertension reaction and aggregate them according to the groups. And then we've used the rule engines to set the queries to be 1 for hypertensive events without the use of aspirin, and 2 the hypertensive events with the use of aspirin. And then we want to view the results. So let's see. Then, so we have here a very high number, or as you can see here, the hypertensive events with the use of bevacizumab alone is much more higher than the hypertensive events when patients use bevacizumab and aspirin. So again, this workflow is very easy to use, and you can just, what is magnificent is that you can visualize the result, and it is very easy. You don't have to own sophisticated computational skills. And when we compare the number of reports extracted, we can see that the Knime has few more cases with less time, around 7 hours, for the total extraction, while again, it took us 1 week to extract all of the files from the dashboard from the years 2015 to 2020. So it is very time efficient. And lastly, despite the advancement in creating a standardized data set or for collecting information on adverse event, but there is still room for advancement. So although the FAERS is an international database, but it only provides a national report. So we wanted to use other databases, international databases, to have a more comprehensive view of adverse events. And the next thing is we want to use other databases because they have a different spelling of medications that may not be captured in databases like FAERS. So this will give us maybe a more global perspective on adverse events. And with that, I end my presentation. Thank you so much, and we welcome any questions.
Thank you, Lama and Sebastian. That was amazing.
Yes, indeed.
And we
I
are
see.
hoping. Sorry, go ahead, Alini.
We already have questions here. We have one by Reza Baharmand. Hi, thank you. I would like to know the mapping you used from Athena. Is it available in Knime Hub?
Hi, Reza. It's not actually available in Knime Hub, but you can open that article that I've shared, and they have all of the information.
And when we were organizing this webinar, we wanted to give you guys two views. One is someone like Sebastian and a whole group trying to tackle a clinical problem. How would they go about doing it? And as Reza will answer your question in a second. But how would they go about doing this? And you can see that you can brainstorm on a whiteboard and then translate it into a Knime workflow. Then with Llama, it's like, how would one expert take a project all the way from concept to implement, from idea to concept, developing the concepts and final implementation. The point I want to make is that both those are highly amenable to Knime because of the way it is built.
Thanks, Shanaka. We have another question here, also by Reza. I'd like to know, did you get the mapping standard from CTM or HGSI?
Yeah, yes, that's yes.
Okay. Do we have any questions for Sebastian? We still have a question here to Lama. I have a question for Sebastian, but let's wait. Are you planning to commercialize the tool from Llama? Is that possible to commercialize it using Knime?
I'll leave. Yeah.
No, we
I'll
don't.
leave
We don't
that
have
to
any
Dr.
plan.
Shanaka.
We don't have. Knime doesn't have any plan to commercialize this tool.
Yeah, we everything that we build in the lab, we try to release it under an MIT license, in the hope that it will end up saving lives. If somebody wishes to commercialize it, Knime is a use case. It's good at what it does, but is the ultimate goal to create a global product that can be used by everyone? We only use Knime to showcase a use case and how you can go from end to end and create the actual logic behind it. So this is Llama's master's project. Actually, she owns, because it's copyrighted, Llama owns everything there. So if you guys want to get that copyright, you'll have to pay Llama.
Thank you.
Yeah, and I let students commercialize things that they it's free. They can go and commercialize it as long as they don't prevent us from continuing development and continuing the science.
Do we have any questions for Sebastian? If not, I have one. Sebastian, have you considered using data to get specific parameter values for your heparin application that can lead to different insights? Like how do these parameter values, how did you get to those values above 150, for example? What's the reasoning behind it?
Yeah, so basically all of those parameters that are set within the heparinomogram are determined by clinical studies. Also, two, it's determined by the actual drug manufacturer in terms of their drug information. And it's basically all of these hospitals determine these parameters in order to kind of assess their own safety. Each hospital has their own kind of parameter settings. All of these can be adjusted per hospital. We just use VCUs since like that's where we are. But all of those kind of like 40 to 45 or 54 kind of ABTT ranges, and then like all of those parameters are set to what the nomogram is. However, in terms of like data collection, like you can if there was implementation of Knime with Epic, which is kind of the EHR system that we use at the hospital, you could link up all of the values that automatically pop up from just regular values that are obtained through blood tests, and automatically set something up that kind of is almost like a warning or almost like a quick side panel calculator, where it just grabs data, processes it automatically, and then automatically, pops up. But because we implemented everything through Knime, we kind of had to manually set those parameters based off the nomogram and then input all the data. I hope that answers that question.
It does. Thank you. There is
course.
another question here that can be answered by you, by Shanaka, by Lama. Do you think Knime could be used similarly for medical devices as well?
Let me take that one, Alini. Anytime you have data, the biggest challenge is creating a prototype that you can test, right? With Knime, you have the ability to put it into production for sure. But for us, the initial value is developing and testing that prototype. So if we have a medical device and we need to test how to translate the data from the medical device into clinical insights, Knime is the best way to go. Once that part is done, you can put Knime directly into production, and it's pretty straightforward to do. But then it's also worthwhile having a conversation with your IT team, whether do you want to put Knime into production or do you want to do something else. But that's a subsequent conversation. Getting that proof of concept done and demonstrating value to the idea that you have is where we have primarily used Knime.
Great. Yeah, there are other use cases in the booklet even that involve all the medical devices, all the medical applications. Thanks, very interesting approach. But Sebastian, would a hospital allow you to use this workflow for real case scenarios? I mean, from the regulation point of the hospital, Vladimir asked this one.
Yeah, I do believe that the hospital would actually allow this, and we actually had conversations with the hospitals to implementing this. It was just the bottleneck was just the linking between the Epic system and also Knime. However, it has to go through two phases. One, it has to go clinical validation. So it has to be validated by the actual clinicians and the hospital board. And two, there has to be kind of like the whole regulation portion of being able to implement, Knime to be able to be used with kind of sensitive patient information and all of that stuff. Make sure that encryption is put in. But you can validate this within hospital systems, and once that validation is actually put through, then you are able to, in theory, use it in the hospital system.
And to add to that, Vladimir, we've been playing around with creating a unidirectional HL7 connection to Knime. In the sense, we are not going to be writing back into the EPIC system, but once you create a prototype like that, how do you do side-by-side testing? So if there's a unidirectional HL7 that we can use as a data ingestion system that goes through the data comes in, goes through the Knime pipeline, and here are the recommendations. Recommendations are not to be used, but to test side-by-side over a period of years. All right, this Knime-based recommendation and the traditional practice, which one is better? And once you understand that, this is like testing it in the clinical setting, right? Once you understand that, then it's a different conversation you need to have with the hospital. Okay, will you allow Knime in here, or would you want to take the logic behind this workflow and implement it into EPIC in their own native format?
Okay. Thanks, Shanaka and Sebastian. There is a last question here by Reza to Lama. If you allow me, last question. Do you have any idea to bring CDM or HGSI or Atlas to Knime?
So we used the CDM OHDCI, but we only downloaded the files from the Athena. So we loaded the files and then we uploaded them into Knime. So this is the only thing you can do, you need to do.
Thanks, Lama. So we're going to stop taking questions for now. I would like to share a little bit of information before we close our webinar. I see that someone posted a question here. Yeah, no, not a comment. I would support the use of Knime for prototyping. We have recently used it during the data exploration and analysis phases of a survey. Thanks, Lawrence. We really appreciate that. So before we go, I would like to quickly, first of all, thank Sebastian and Lama very, very much, and also Professor Shanaka for, this incredible collaboration. And what I would like to do before we close is to quickly share that we are having a summit this spring in Austin, Texas. So from April 15 to April 17, it's a big event. You can join online for free. There is a virtual summit pass, or you can get the on-site summit pass that gives you the access to the event, the 16th and the 17th. And this costs $350. And you can also get access to the whole event, the 3 days, including some training. We have lots of courses on different aspects of Knime, and this costs $650. You can expect not only these training sessions, but also a lot of networking, workshop, birds of a feather, partner exhibitions, online breakouts. And why am I talking about this summit here? Not only because it is a great event, the most important event for the Knime community that is happening this year in the US, but also because Professor Shanaka will be there, not only as a panelist, along with other professors and industry practitioners, but also signing free copies of his booklet that he co-organized with Ali Marvi, who works for Knime. So I say his booklet, but as you can see, it's a booklet that he organized, but actually involves use cases from many different students, including Sebastian and Lama. And you can download this book for free by going to knime.com/knimepress/boosting digital health. But if you want an actual physical copy signed by Shanaka, he will be at the summit also doing that. So I would like to use this final moments to thank you very much for being here. And it would be great if we could meet again at the summit or in any other opportunity. Thanks for being interested in Knime, for using Knime for prototyping, and for being interested in learning more about Knime has been used for digital health. Thank you very much.